To understand how MR spectroscopy works, we must look past the macrostructures of the brain, zooming down to an isolated, microscopic cube of tissue. Standard MRI uses the vast abundance of water to map spatial anatomy. MR spectroscopy isolates specific metabolites to map biochemistry. While we can target carbon-13 or phosphorus-31, we almost entirely rely on hydrogen-1, or protons. We target protons for three distinct reasons: 100% isotope availability, high magnetic sensitivity, and abundant presence inside target metabolites. We are repurposing an anatomical camera to act as a highly localized biochemical assay. Protons existing in different molecular environments experience microscopic variations in their local magnetic field. Because that local magnetic field changes, the Larmor frequency of those specific protons also shifts by a tiny margin. Isolating molecules reveals their proton groups each emit a distinct frequency wave. This chart measures microscopic shifts, collapsing the waves and plotting them horizontally in parts per million, or ppm. The vertical axis measures peak amplitude, indicating exact signal strength. A higher signal strength mathematically equals a higher concentration of that specific metabolite in the tissue. By charting these frequency shifts against signal strength, we extract a precise spectral signature that reveals the exact chemical makeup of the tissue voxel. This graph illustrates the clinical metabolite spectrum, starting with a single, bold central peak at the 3.0 mark. This is creatine, a marker for energy metabolism. Because it remains remarkably stable, creatine acts as the constant marker against which we compare every other peak. To the right, at 2.0 ppm, is N-acetyl aspartate, or NAA. This is our primary marker for healthy neurons. A drop in NAA indicates conditions resulting in neuronal loss, while a sharp rise is a specific indicator of Canavan's disease. Over at 3.2 ppm, we find choline, a marker for cell membrane turnover and metabolism. When this choline peak elevates, it acts as a primary indicator for tumors and demyelinating diseases. Further down the spectrum, at 1.3 ppm, we have lactate, identifying anaerobic respiration. A raised lactate doublet signals underlying issues like ischemia, seizures, tumors, or mitochondrial disorders. At the far right of the graph, between 0.9 and 1.4, we see markers for profound tissue destruction. Lipids indicate necrotic tumors, while amino acids point to pyogenic abscesses. We also track supplementary markers like myoinositol at 3.6 ppm for glial cells, and GLX between 2.1 and 2.5 for neurotransmitters. Interpreting this specific arrangement of peaks allows a radiologist to differentiate between healthy brain tissue, an ischemic stroke, or a necrotic tumor using purely mathematical physics. To actually capture this data, we have to solve a core signal-to-noise problem, extracting trace metabolite signals from a biological system composed overwhelmingly of water. The immense hydrogen signal from water completely buries our vital metabolite data. Step 1 is water suppression. A chemical shift selective, or chess sequence, sends an inverse wave, flattening the water signal to zero, revealing the pristine metabolite peaks underneath. Step 2 is voxel selection. We must isolate exactly which piece of tissue we are extracting the spectrum from. In single voxel spectroscopy, we use successive RF pulses in three orthogonal planes. Where the X, Y, and Z axes perfectly intersect, a single, isolated voxel is targeted. This method provides a high signal-to-noise ratio and fast scan times, but it only covers a very small area. To evaluate a wider region, we use multivoxel chemical shift imaging, which utilizes phase encoding steps to map a large 3D grid. This offers larger total coverage and higher resolution. However, it requires longer imaging time and introduces the risk of spectral contamination from adjacent voxels. Step 3 is spectrum acquisition. We run specialized sequences like press or steam to record the final spectrum. Without executing this exact triad, suppressing water, precisely isolating the voxel, and running a specialized acquisition sequence, the critical metabolite data would remain invisible. Despite its chemical precision, MR spectroscopy is inherently low resolution compared to standard anatomical MRI. It is also extremely vulnerable to local magnetic field heterogeneity. Imaging close to bone, calcium deposits, or flowing blood disrupts the uniform magnetic field lines passing through the tissue. These physical distortions ruin the subtle Larmor frequency shifts we rely on, causing severe artifacts in the final spectrum. To correct these local distortions, the machine requires a mechanical countermeasure called shimming, which actively homogenizes the magnetic field. By mastering Larmor frequency shifts, executing chest suppression, and interpreting these specific spectral markers, radiologists can diagnose complex pathology at the molecular level long before structural anatomy ever changes.